So hello everyone, welcome to our webinar Overcoming Challenging in Single-Cell Sequencing Sample Preparation. And my name is Xiaohui. I'm Scientific Application Manager of Novogene EMEA. So join us today is Caroline from Genomics and Dr. Xing Wen from Miltenyi. So there are some housekeeping items. So after this presentation, we have a Q&A session to address the question that's submitted by our audience. And because the time limited, we may not have chance to answer all the questions, but no worries. Our team will reach out to you later. So yeah, I will start by briefly introduce Novogene as a company, so everybody on the same page. So our Novogene is founded in 2011. So this year is our 10 years anniversary. Our mission and the goal is to advance genomics to improve human health and life. So you can see this slide here. We offer a lot of end-to-end sequencing services cover like whole genome sequencing, whole transcriptome, epigenomics, metagenomics, oncology, and a lot of other we didn't list here. And we use the most cutting-edge sequencing platform like from Illumina, PacBio, NanoPower, and Transgenomics. So this is show our global presence. We have 5 labs around this globe and many offices around all these countries. So I want to highlight here is our Novogene EMEA is headquartered in Singapore. So we have a lot of office in this EMEA region, like in countries like Japan, Australia, New Zealand, India, and Africa. Yeah. We also work with local partners to provide the sequencing services to our valued customer. So thanks to the hard working from our employees, and also appreciate our customers. And the customer using our sequencing services has made a lot of progress in their scientific research and published tons of papers on the top journals like shown here. And you also can see we collaborate a lot of like more than thousands of hospitals, research institutes, and the company around the globe. So now I will start the introduction of our single-cell sequencing services. Yeah. So before start, so everybody need to know what's the goal of single-cell sequencing services. I'm quite sure not everybody knows what's the goal of the single-cell sequencing services because many are very new to this very new technology. So the single-cell sequencing is to measure the gene that are being expressed in each individual cells. And this technology is very popular and a powerful technology that enable biological questions that cannot be answered by this so-called bulk RNA sequencing experiment. And shown here is just like how this single-cell RNA experiment works. So all the single-cell experiment starts with this dissociation of the solid tissue into single cells. And after removing this dead cell, the single cell can be input into this droplet system for single cell capture and the library prep. So the sample dissociation into the single cell is some scientists comment as the most challenging part. So later, experts from Genomics and Miltenyi, they will dive into this sample preparation part. And shown here, this is two-cell or separation system FACS, the method can be used to enrich your target cell population. And so how this cell capture works, the idea is to capture the cell shown here and the beads, very teeny tiny beads into this droplet. And inside the droplet cell will be released and mRNA will be released and captured by these capture oligos on these teeny tiny beads. And these capture oligos have cell barcode and UMI. UMI means unique molecular identifier. So cell barcode will allow the downstream data analysis to map the NGS risk back to this particular cell. So you'll know where the risk came from. And The downstream is just to very traditional molecular biology work to convert the mRNA into cDNA and incorporate this cell barcode and the UMI, and followed by sequencing and demultiplexing and data analysis. So many are interested. I think many researchers are interested what kind of research questions can be answered by this single cell experiment. So this one, I will show some examples here. Like this one is the most popular application of single cell sequencing in profiling the known and the novel cell types, like in different species. So shown here are 3 examples. This one is the mouse small intestine. They identify 15 cell populations, and this one is to identify the population in heterogeneous the Arabidopsis root tissue. You can say many population identified, and this one is to identify this heterogeneous population in mouse mammary gland. So another very powerful application of this single cell is to infer the trajectory to understand the development, differentiation, and how disease development by reconstruct, they call the developmental trajectory or differentiation trajectory to understand how cell fate is determined. So this first example is to identify how cell differentiate to form this luminal compartment in mammary gland, and also use this single cell experiment to identify a common progenitor. Then this progenitor will follow these 2 paths to differentiate into these 2 different subpopulations. And during this differentiation, different gene expression is switch on or switch off. And so here is another example. This one is study of OSN. It's called olfactory sensing nerve cells. They also identify reconstruct a linear trajectory. The progenitor will start from here and differentiate into a middle stage, immature OSN cells, and finally differentiate into the mature OSN cells. So this one is this RB root cells, so different from very early stage to the late mature differentiated cells. This one uses different. And also single cell is hiding into this so-called precision medicine. So many publication is focusing like how so-called this immune checkpoint inhibitor working at single cell level. And it's like single cell sequencing can be used as a biomarker to predict the response of patients to this checkpoint inhibitor. And what's the mechanism of this drug resistance? Is that question can be answered by single cell sequencing. So for this paper also identify these 2 cell states is for CD8 positive T cell that respond to this therapy treatment, to this inhibitor treatment, and CD8B that not respond to this treatment. So they identify these 2 cell states and different marker associated with this cell population. Then they also even use this inhibitor targeting this non-responder gene expression markers for the improved treatment. So here I want to mention is cell multiplexing techniques that like scientists are working very hard. Like almost every day, that new technology, new tools come out to either analyze data or to perform the experimental in a better optimized way. So this is the one I want to mention, cell multiplexing. So this one is just use the algorithm to demultiplex the data into these individual cells. So it's only suitable for human samples. So you can put the sample together without prior knowledge of this genotype. Then this algorithm can infer this genotype based on this common variance in the population from like Southern Human Genome Project, and the risk can be identified, assigned to these individual cells. Yeah, it's really suitable for the human subject studies and dramatically reduce the cost associated with the single cell level population. Yeah. So shown here is another technology, has technology. So this technology, you can label the cell or cell memory or nuclear memory with this hashtag, and then do the cell capture using this 10x genomics platform and do all this downstream. And this technology has been commercialized by 10x Genomics. So using the 10x Genomics cell plus kit, scientists can pull like 12 samples together and load into this channel for single cell library preparation. So our services, we use this 10x Genomics platform for single cell sequencing. So shown here is this cell capture platform. We use this Chromium controller, and shown here is the chip for this gene expression. And the chip has 8 channels, so can run 8 samples within 18 minutes. So Cartoon just show how this actual droplet formation. And on the right side is library prep. It's again very traditional molecular biology work. So once the library ready, we will our sequencing team will perform the sequence to generate this BCL file, and we use this fantastic cell software from 10x Genomics to do this data demodelizing, and then run cell ranger report to check the metric, like how many cell capture for each experiment, how many read sequence, how many genes identified, and also this sequencing and the mapping matrix. And also cell ranger will generate a file, it's called gene expression by cell matrix. And this matrix file can be loaded into the downstream like Surat or any Scampi software for the bioinformatics analysis. So this is, we also provide this bioinformatics analysis, like after we got the gene expression by cell matrix, we'll input this software like Surat and Scampi, and we do this cluster analysis, and this cluster will be used for downstream like cell composition analysis, pseudo-time analysis, differential gene identification, and a pathway annotation. Yeah. And a lot of more customized analysis I didn't show. So shifting from the traditional bulk tissue RNA-seq to single-cell experiment is really a very big shift in the whole field. So like there are many challenges to enable a successful single-cell experiment. So first one, like shown here, is the cost is very still prohibitive, and the sample prep is really challenging. And also for data analysis, the individual lab, maybe they do not have enough big server to do this data analysis, because there's a really huge amount of data. So to overcome this treatment challenge, to make like the single cell sequencing technology can be accessible by each individual lab and each scientist. In the beginning of this year, we launched this single cell sequencing services. Yeah. And recently, we also have a very attractive offer. So you can see here for single cell gene expression library prep, each library will only charge like less than 2,000 Singapore dollars. And also for FAP gene expression, we charge like just $3,000 plus. And we also have an attractive offer for the spatial transcriptomics. Each library prep, we just charge less than $9,000. So we also offer this DIY use, like if you already have reagents, instrument, all this, then you do not have a controller. We also can logistic the controller to your lab, so you can perform this cell capture in the library preparation. Yeah. And also for all these details, you please reach out to our sales representative. And so here is like how our service looks like. We provide this end-to-end single-cell RNA services. So our service starts from this project consultation. So if you are interested in single-cell sequencing, please talk to our commercial team and our single-cell expert. And to know more about like how many cells you should capture, how many reads you should sequence for each cell, like how to optimize this single cell suspension preparation. So we can provide all this technical support. And once your single cell or single nuclei suspension is ready, we can collect this single cell or single nuclei suspension from your lab. And also we can move our instrument to your lab to perform this cell capture, library prep, and we can do all this downstream sequencing and this data analysis. So here I just do a very brief breakdown why you should test the single cells sequencing services. So this is just comparison. So if you own your system, you need to consider all this maintenance for this platform, and also need to buy all these other essential instrument, reagents, consumables for the single cell experiment, and also need to consider the manpower for this system running. And also this is the very general cell isolation library prep. And I know that every time, not every lab, they have a Novogene sequencer. You need to consider where to outsource the sequencing. And some lab, they may not have the expertise in this bioinformatic analysis. And here is shown what our single cell sequencing services was the advantage. So if you use our services, you don't need to consider about that maintenance, all this whole platform. And no need to buy all these smaller instrument reagents to run the experiment. And we have expert to run the cell culture and all this experiment for you. Yeah. And we also can like provide this cell isolation support and do all this downstream library prep sequencing and bioinformatic analysis. Yeah. So I just want to highlight again here that we provide these on-site services. So if you have a cell, single cell or single nuclei suspension ready, just let us know. We can do cell capture, library prep anytime, anywhere. Yeah. So this will really dramatically reduce in your lab workload and capital investment, enhance the scientist's ability to exploit the power of single cell sequencing technology and accelerate this scientific discovery. Yeah. So this is our commercial team across this EMEA region. Yeah, so please reach out to our team to discuss more about your single cell sequencing experiment. And also, to mention here, we also have a lot of other very attractive offers. So this one is the RNA stable tube. So for RNA sequencing, most of the researcher will worry about like I need to do the dry ice treatment, otherwise my RNA will be degraded. So we offer this, this magic tube that you can store the RNA in this tube. Then this tube you can ship at room temperature. RNA will be very, very, very stable. And we already have a lot of testing. It's very good quality. Yeah. And we also offer this free RNA extraction services for all this RNA sequencing experiment. And we also offer bacterial DNA extraction, more than 40%. So if you are interested or you plan to do any RNA-seq, bulk RNA-seq experiment, please reach out to us, commercial team, to learn more about details. Yeah. So yeah, finally, you can also visit our virtual booth to learn more about our genomic solutions. We really provide a lot of genomic sequencing solutions. Yeah, thanks for your time. So now let me introduce our first speaker, Caroline Chang. Hey, Caroline, how are you doing?
Good.
Yeah. Let me briefly introduce Caroline. So Caroline received her PhD from University of Queensland in development biology, where her research focused on olfactory system growth and regeneration. So after her PhD, she has joined the industry, working with leading international and macro and life science company. And currently, Caroline is staff application scientist at Genomics in Singapore. Thanks, Caroline. I think you can share your slide now.
Okay, great. Let me share my slide. Okay, can you see my screen?
Yes, we can see your screen.
Okay, good afternoon, everyone. Thank you very much for attending the webinar today. So in this session, we're going to talk about sample preparation guidelines that 10x Genomics have for doing single-cell gene expression analysis. So just a brief overview of how the next-gen Chromium single-cell gene expression solution with the feature barcoding technology works, right? Xiaohui has mentioned briefly about it, right? So basically, three components are bring together, right? They are your single-cell suspension that are mixed together with the master mix, consisting of the reverse transcriptase enzymes and the reagents for, to carry out the reverse transcription step, and the 10x barcoded gel beads, where each of these beads contain its unique 10x barcode and the partitioning oil. So these are placed on the Chromium chip and then placed on the Chromium controller, or we have recently launched our high throughput instrument called the Chromium X system to generate what we call the single cell gems or gel bead in emulsion. So within the gem, there's possibility that some of the gems do not contain any cells, but just the 10x barcoded gel beads, and then some of the gems consist of a single cell together with the 10x barcoded gel beads. Then once the gems are formed, you then carry out the reverse transcription reaction to create the 10x barcoded cDNA, and once we have the 10x barcoded cDNA, we can break the gems and pull the barcoded cDNA together for downstream library construction. So what is sample preparation? Sample preparation is defined as how the sample is being treated prior to analysis. So this, of course, involves the physical sample that you bring to the experiment. And in this case, for single cell gene expression assay, it will be either single cell suspension or single nuclei suspension. So the whole sample preparation workflow require a set of decisions from the planning stage, like the types of samples that you are going to analyze, to the sample preparation optimization steps, and the final eventual sample run. And of course, it involves how you're going to determine the quality of your final cell suspension. So the input material for the next-gen single-cell gene expression assay is your single-cell suspension or single-nucleus suspension. So, and this starting material, the quality of the starting material could be impacted very early in the workflow. So there are a few questions that one needs to consider. For example, what are the sample source, right, where the cells are extracted from, and then how are the samples treated prior to arriving in your labs, and are there any specific considerations about these tissue types or sample type that you're working with? How much do you know about the starting material? Are the starting sample tissues, or are they self-suspension? So if they are tissue samples, then you need to dissociate the tissues to get your single cell suspension. So successful sample preparation required a set of decisions that dictates the best workflow for preparing your cell suspension. So it involves planned early, right? Start your planning, right? For example, you want to decide on the sample type that you're working with, and then review appropriate demonstrated protocols, either from the 10x Genomics support website, or looking for references that have used a similar tissue types that you're hoping to work with, and then develop a workflow plan for this particular sample type. And in terms of the sample preparation optimization, of course, you would need to evaluate dissociation protocol if you're working with tissues, for example, or any enrichment step required to enrich for your cell type of interest, the storage and transportation of your samples before you actually start the actual single cell experiment. You would want to assess for the viability and morphology of your cell suspension to make sure they are highly viable, and then the cells look healthy, and also with the cell suspension to whether any additional cleanup steps are required to remove cell clumps and debris. And if you are working with our feature barcoding technology on top of the gene expression profiling, you would also need, for example, looking at cell surface protein detection, you would also need to optimize on the cell surface labeling. And finally, in the final actual sample run, then you want to use the optimized cell protocol that you have already done beforehand. Treat your samples gently and work quickly and efficiently during your sample preparation steps, and make sure that you determine an accurate cell concentration so that you do not underload or overload the system. So the key to a successful single cell experiment is really the quality of your single cell suspension. What we really wanted is a clean, healthy, and cell suspension. So we want a clean cell suspension that is free of aggregates and clumps. The cells are healthy and they are intact. They are highly viable, right, without much physical decomposition that cause lysis of the cells, which tends to release ambient RNA to the suspension buffer, and that will reduce the overall quality metrics of your data set. So one of the useful website for the sample preparation resources is the 10x Genomics Support website. So if you go to our 10x Genomics Support website and you search for single cell gene expression application, for example, this website consists of documents for the general sample preparation guideline, specific demonstrated protocol for different types of tissues, and also if you're working with the feature barcoding technology, we do have the cell surface protein labeling protocol and the CRISPR screening guideline as well. Okay. So the cell preparation guide is one of the documents that is available on the 10x Genomics Support website. This guideline provide a lot of best practices and general protocol for cell washing and resuspension, cell straining, cell counting, optimal cell concentration, as well as recommendation on how to work with samples with low input number of cells, which is less than 100,000 total cells. And in general, flow sorted and primary white blood cells can be treated like suspension cell lines. So when you are planning and optimizing your sample preparation workflow, there are a number of points to take note, right, from your sample procurement to tissue dissociation if you're working with tissues, whether you are doing any enrichment or separation steps once you have a cell suspension, to the storage and transportation of your cell suspension, and of course the final cell count and the quality check of your final cell suspension. So all these steps need to be optimized prior to start the actual single cell gene expression workflow, and in order to maximize the quality of your final cell suspension, you want to treat your cells gently during the preparation of your samples so that you do not lyse the cells and increase the release of the ambient background RNA into the environment. You want to make sure that you work quickly and efficiently when you're preparing your samples and count your cells accurately to check for cell debris and aggregates that's present in the cell suspension as well. So if you are working with the 3' gene expression assay, right, and if you're working with cultured cells and PBMC, we do have a pretty well-established demonstrated protocol for cultured cells and PBMC, where there should be a minimal optimization required for this type of samples. And for fresh tissues, dependent on the actual tissue type that you're working with, there might be some optimization required in terms of the tissue dissociation. And the assay works also with frozen tissue, but with frozen tissue, you do need to extract the nuclei instead of the cells. So it will be technically slightly more challenging compared to doing single cell suspension. And if you're working with fixed cells, we do have a methanol fixation protocol that is possible with the single cell gene expression assay, but we do prefer cryopreserved cells over methanol fixed cells. So shown here on your right, basically the types of starting material that is compatible with the assay, the 3' gene expression assay that we have. So if you are interested in the 3' gene expression assay, your starting material could be fresh cells or tissues, it could be cryopreserved cells, it could be nuclei from fresh tissue or frozen tissues, as well as methanol fixed cells. And if you're interested in doing cell multiplexing, we have also recently launched our CellPlex reagent, which allows you to actually multiplex the number of samples that you can load per channel on the system. So with the cell multiplexing protocol, it is compatible with fresh cells and tissues, cryopreserved cells, nuclei from fresh tissues, but it is not compatible with snap frozen tissues, nuclei extracted from snap frozen tissue and methanol fixed cells. And with the feature barcoding technology on top of the gene expression assay, which is CRISPR screening or cell surface protein detection, it is only compatible with fresh cells from fresh cells, fresh tissues, and cryopreserved cells. So shown here are the routinely used demonstrator protocol available on the 10x Genomic support website that we have for single-cell RNA sequencing. So we do have the optimized protocol for cell lines, mouse embryonic neural tissues protocol, as well as PBMC, which consists guidelines on how to freeze your PBMC and thaw the PBMC. So in terms of cell handling, as mentioned before, right, the key to the successful single-cell experiment is really a clean, fully dissociated single-cell suspension. You want to minimize the presence of cellular aggregates, dead cells, and also potential inhibitors of the reverse transcription reactions, such as high EDTA or magnesium content. And then in terms of cell viability, right, we do recommend to try to achieve a cell viability of above 90%. Of course, this is not a clear cutoff, right? Doesn't mean that you do not get a 90% viability of your cells. You cannot carry out the single cell gene expression assay. It is just a current recommendation, and I would say that minimally you want to try to achieve 70%. If you have a cell suspension with low viability, then you can make use of, for example, like the dead cell removal kit from Miltenyi to try to improve your cell viability. Then the key and the reason for that is because we want to prevent the presence of a high fraction of non-viable or dying cells that can release ambient RNA to the environment. And this will impact the quality metrics reported by the software. So when you are treating and handling your cells, right, you want to treat your cells gently to minimize cell lysis and cell loss, right? So you want to wash your cells with white ball tips and wash and resuspend the cells in PBS with 0.04% non-acetylated BSA. And for debris and aggregate removal, for example, if you're doing like tissue dissociations and it tends to like generate a lot more debris with the dissociation protocol, then you could use a cell strainer to remove the aggregates or debris from the suspension, right? And the reasons that we need to remove the cell clumps and the debris is that it could potentially clog the channel of the instrument if the clumps is too big, right? The size of the channel is about 50 to 60 micron. So if you have like big cell debris or large clump, it could potentially clog the channels during the gem generation. And then we want to make sure also that the cells cell counts are counted accurately before loading onto the system. The optimized cell concentration that one try to achieve is from 700 to 1,200 cells per microliter. And of course, with the standard kit on the Chromium controller, you can recover somewhere from 500 to 10,000 cells. And if you're doing the cell multiplexing protocol, you can recover up to 30,000 cells per channel. And with the newly launched Chromium X system and the high throughput kit, you can recover up to 20,000 cells per channel. And if you're doing cell multiplexing, you can recover up to 60,000 cells per channel with the Chromium X system. So you want to make sure that you count your cells accurately so that you do not potentially overload the system with more than what you expected. And also, or if you undercount your cells, then you underload the system with less cells being recovered. And storage of your single cell suspension, generally they are stored kept on ice. And then we do recommend to keep the time on ice as minimal as possible, right? Depending on how fragile the cell type is, right? Some cells, they do survive very long by keeping them on ice. So this will also be something that you need to consider during the optimization protocol to check the viability of the cells before you load the cell suspension onto the system. So shown here are some of the cell types that has been validated in-house at 10x Genomics. So you could see that the assay works well with a wide variety of cells containing a different content of RNA, from low RNA content such as PBMC to higher RNA content of cell lines. And the cell size that we have tested in-house is up to 30 microns, but we do know that it does work with greater than 30 microns. So technically, if it's less than 50 to 60 microns, it should work as well, as we know that the channel size is about 50 to 60 microns. So here in this table here, we show the data with how you pipette the cells, that how it affects one of the QC metrics of your data set, which is the fraction reads in cells. So you could see that comparing between control using white ball gentle pipetting and then using the white ball tips with rough pipetting versus using the narrow ball tips with rough pipetting and vortexing your cell suspension for 5 seconds. So we see that using white ball tips with gentle and rough pipetting, the fraction reads in cells are still pretty high, above 70%, but comparing that using narrow ball with rough pipetting, where you tend to maybe generate a lot more air bubbles when you're pipetting, there are a lot more cell lysis created, and that is actually worse than vortexing your cell suspension for 5 seconds. So it is important that when you're preparing your cell suspension, make sure that you do gentle pipette and then try to use white ball tips during that step. So as mentioned before, we do have a well-established protocol for PBMC, and in this particular protocol, we have the recommendation on how to do cryopreservation and also the thawing protocol for these types of starting material. And the assay also works well with cell lines. And then one thing to consider is the time that you need to dissociate the cell from the cultured surface. So the incubation times might vary depending on the cell line that you are using. So some minimal optimization might be required on the dissociation time. So our single cell gene expression assay has been demonstrated and worked pretty well with cultured cell lines with using enzymatic dissociation of all the enzymes indicated here. It works also well with primary cells such as solid cells, magnetic bead purified or gradient purified cells. And if you're working with solid tissues, you need to work out the best tissue dissociation protocols, either from looking at the demonstrated protocol available from our website, or the other alternative database that provide a lot of useful resources is Worthington database, where there are a lot of publications with different types of tissues that other customers have worked with, and this will be a useful resources to find the dissociation protocol of your tissue types of interest. And if you're working with solid tissue, you could use enzymatic dissociation with fresh tissues, or if you're working with frozen tissue, then most likely you need to use mechanical dissociation. So in terms of cell washing, we recommend to wash your cells twice to remove ambient RNA and any potential like debris, right? So shown here on your right, the graph shows the cell viability on the y-axis versus the number of washes that you have done to your cell suspension. So you see that washing the cell twice or 3 times, the cell viability are fairly similar. So we do recommend to wash your cells 2 times to get rid of the ambient RNA as much as possible. But of course, if you're working with a very low concentration of cell suspension, it is possible to reduce the cell washing steps to 1 time to minimize further cell loss. Some of the validated in-house buffer and media is shown here, right? For example, the commonly used HBSS or PBS and the cultured medium for the different types of cell lines. These are the buffer that we have validated in-house. So sometimes you do need to consider what are the additional cleanup steps or enrichment steps that you need to be carried out with your sample of interest. For example, you want to do additional washes to get rid of the ambient RNA and debris. And you need to maybe do filtering, like using cell strainer to get rid of large clumps, or are you interested in enriching certain population of interest or depleting cells that you do not want to investigate? So these are something that you need to consider with the sample type that you're working with, and optimization might be required in these steps as well. So for debris removal, we do recommend to use cell strainer from the Flomi cell strainer from Bell Art or the Mac Smart strainer from Miltenyi Biotech. And you could see the image at the bottom, right on your left is the unfiltered cells, and then in the middle and on the right are the filtered cells with Flomi cell strainer and the Mac cell strainer. So you see that after filtering, the big clumps are removed, and then the cells are more in a singulated cell suspension. So when working with tissue starting material, right, so then you need to dissociate the tissues to obtain your cell suspension. So there are two options, either you're working with fresh tissues or frozen tissues. So if you're working with fresh tissues, you can use the enzymatic dissociation to get your single cell suspension. And it is also possible to work with nuclei suspension if you're working with tissue samples. But generally, you would probably want to work with single cell suspension because they are easier to prepare compared to isolating nuclei. But in cases where you have cell types that are big in size or irregular shapes, then you might need to actually extract the nuclei instead of preparing single cell suspension. And for frozen tissues, you do need to do mechanical aggregations, right? And then throughout all this dissociation process, there will be a QC check and in the workflow to make sure that you have a clean cell suspension or nuclei suspension. Okay, in the last part of my talk, I would like to also give some guidelines and resources that we have for nuclei isolation for single cell sequencing. So we do have the nuclei isolation demonstrated protocol for single cell sequencing on our 10x support website, right? So we have the nuclei isolation from cell suspension and tissues, as well as the adult and neuronal mouse brain tissues for the nuclei isolation protocol. So these are some of the demonstrated protocol that we have worked in-house and that you can use as a reference to the sample types that you are working with, and then try out whether this works well with the tissue samples that you're working with. And the other commonly used customer developed protocol is called the Frankenstein protocol for nuclei isolation from fresh and frozen tissue, developed by Luciano Group. This customer developed protocol has been commonly used by many different customers to isolate nuclei from fresh and frozen tissues. So this will be a useful resource for people who are interested to do nuclei isolation from a tissue sample. So in terms of viability, when you're extracting nuclei for your single cell sequencing, of course, then the lysis, the cells will be dead. Most of the cells will be dead, right, when you're extracting the nuclei. So you're expecting that above 95% of the cells are dead, and then less than 5% of live cells when you're extracting the nuclei from these tissues. And besides checking the cell viability to make sure that you have low viability and then free of cell clumps, it is also very important to check the nuclear morphology under the bright field microscope. So we do recommend to check the morphology of the nucleus using a 60x magnification, but 40x is also possible to check to use to check the nuclei quality. So what we wanted is really something you see in image A, where you see a high quality nuclei with well resolved edges, right, very smooth nucleus membrane, and this is optimal for both the single cell sequencing as well as the ATAC libraries. And then if you see some minor blabberings like what you see in image B, right, so this is still good enough to go ahead with the experiment. But if your nuclei quality is something seen shown here in image C and D, where you see strong evidence of blabbing, and in image D you can see that the nuclei are no longer intact. So this suspension will not be something that you want to go ahead with your experiment. So again, on the 10x Genomics Support website, we do have a lot of useful user guide, product sheet, technical notes, as well as how-to videos on sample preparation and on the assay itself, right? So this will be a useful website that you should visit to as a start to get information. And lastly, of course, if you have any questions regarding our products and also questions that you have for your single cell assay, please feel free to drop us an email if you can't find our information on our website. Okay, with that, I would like to end my talk, and thank you very much for your attention.
Yeah, thanks, Caroline. That's a very nice presentation. I think everybody now have a very clear image like how to prepare the sample or what's the best practice. Yeah, thank you very much. So our second speaker is Dr. Xin Wing from Miltenyi. Yeah. So hi, Xin. How are you doing? Yeah.
Yeah, hi. So I'm Sam from Miltenyi, but before this, I was actually in a lab that is actually developing a TCR kind of a therapy, and I was actually one of the inventors of the TCR T cell therapy, kind of immunocell therapy kind of a patent as well. So that's actually very interesting. And I also do a lot of single cell genomics works during my postdoctorate as well. So this is actually quite a bit of my forte as well. So today I just want to share with you guys the preparation of cells for your single cell genomic analysis. Okay, can everybody see? Yeah. So there's actually a lot of prerequisites and requirements for a single cell analysis. As you can see from the previous presentation, how you treat your cells is very important before you actually put it into your single cell machine or analysis. Whatever you do to your cell itself will actually affect your analysis ultimately. Yeah. So whatever you put into the analysis, the quality will also be determined on the quality of data that comes out from your analysis or experiment. Yeah, so things that will be based on the quality will be if your sample is debris-free, if these cells are viable, and are these actually single cells. And of course, you want to do it as fast as possible. Your gene expression, when you're doing all your isolation and preparation step, it doesn't get affected. And of course, your cellular composition. And lastly is whatever cell types that you're putting into. It could be from blood, PBMCs, it could be from organs, tumors, tissues. These will be your cells. And of course, you need to know what you are dealing or doing to your sample. And this will actually incur some bias to your data set when you're actually analyzing your data. So please do keep in mind of all your sample preparation before you actually put them into the machine to actually analyze your data, for example. So we do provide at Miltenyi, we do provide workflow solutions for cell separation all the way from tissue or blood to the sample analysis or single cell analysis of any sample you would want to. And we actually do have all this product that is actually compatible with the 10x genomics. So these are already tested in this platform as well to yield very good results as well. So when we look at the tissue storage, we actually provide a tissue storage solution that will allow you to store your tissue at 4 degrees for a prolonged period of time without your tissue or cells expressing stress markers. So this is very useful, especially if you are doing a biopsy or anything that you want to just use fresh tissue, because fresh tissue is the best. So this is something that will be very useful for you to store your tissue all the way to a few days, or even some users actually have said that they use it up to a week and they're still not showing any stress markers. So next is the dissociation of your tissues or organs. So in our case, we actually have the Genomics technology for the tissue dissociation. So it is actually a mix of mechanical destruction and an enzymatic digestion through a machine. So why would you actually want to incorporate these two particular methods? Why can't I just use this mechanical or enzymatic? It's because if you actually have a combination of both, you actually can minimize the time of the enzymatic digestion of the tissue, for example. And on top of that, when you actually minimize the time of enzymatic digestion, you actually do preserve more epitopes on your cell surface, for example, for you to do subsequent analysis, such as one of the single cell analysis, which is also requiring a surface stain as well. So that is something that is important. And of course, not to forget, it also improves the yield of viable cells compared to just mechanical method or enzymatic digestion method. So of course it's faster and it's gentler because it doesn't have a harsh enzymatic digestion of your cell surface proteins. And also it's reproducible as well because it goes through a machine, it's a kit. So everything is already weighed out, for example. Yeah. And of course, when you look at the total number of cells in this experiment, for example, you actually use a manual method versus a Genomics protocol method, you actually see that there's quite a big number of cells that you can actually get from the Genomics dissociation method compared to just the manual methods itself. So this is actually direct cells from the clonomic lamina propria tissue. So next, so you have actually heard about nuclear extraction. So this is when you are working with the nuclei. So when would you actually want to work with nuclei? This would be when you are looking at snap frozen or frozen samples like previously explained. There's also the OCT embedded tissue. And of course, you can also deal with the fresh tissue as well. So these do open up a bigger portion of samples that you can actually use, because OCT samples are very common in the medical field, where they actually preserve and embed tissue or pathological tissues in OCT. And this is where you can actually use this tissue to actually do a single cell and genomics as well to analyze this data. So this is actually really useful. And of course, it's very quick. It's only a 7 minutes protocol. It also is a user-independent method because it's through automation. And of course, this machine does allows the parallel processing of 8 samples. So we actually look at the different samples of either from different organs or tumor, for example, like brain or liver, kidney, for example, and you compare frozen and fresh tissue or sample that you work with, and you look at the RIN number. This is actually a value to actually gauge how well what's the quality of your sample for DNA quality. So you can see that actually there's very minimal differences between the fresh and frozen samples when it's actually processed through this nuclei extraction method. So this is very useful. And next is, of course, the sample cleaning and also the different sample that you can actually use. Yeah, so sample cleaning is actually very important to actually remove cell clumps, aggregates, debris, dead cells, or other cell types like red blood cells as well. So this is actually very useful to actually eliminate to get you very good quality data from single cell analysis. So you can actually remove dead cells from your sample by actually having using the cell removal kit that we have. So this is actually a ready to use microbeads. It's actually very fast and very straightforward. You can actually get to very pure viable cells within 25 minutes, and you can also have a bit of automation if you actually perform it with the Automax Pro. So on the left-hand side over here, you can see this is actually 2 different samples. So one sample on the top and one sample on the bottom. So you can see over here when you actually have cells, there is actually of like 62% viability. So this is the PI of viability stain on the y-axis here, and the x-axis here is the forward scatter. So you can see with a reasonable viability of 62%, this is actually still not a good viability or quality of cells to put through the machine. So of course, if you actually put it through the dead cell removal kit, you'll be able to get up to like 96% or over 90% kind of a purity of viable cells. And if you actually get very terrible samples, and you get about 37 or 38% of viable cells, when you actually put through these cells or sample through the kit, you can also see that you do get quite a bit of enrichment of your live cells. So you do remove quite a bit of your dead cells also as well. So for very sensitive cells such as your embryonic neurons cells, we actually have the debris removal solution instead. So this is instead of processing through the beads and columns, you can actually just use it as a density gradient reagent, and you can apply full break and acceleration in this particular centrifugation step, and it will actually save you a lot of time and give you a very good quality of sample at the end. So of course, as I was saying, lastly is actually no, not lastly, but next is actually the cell isolation or sorting. So you actually want to ask why cell isolation or enrichment or sorting to get a pure population. Why can't I just have a bulk? So if you actually have particular subset of cells that you want to look at, you'll be able to get directly these data from this particular cells. But however, if you put your cells via a bulk kind of sample, then your cell of interest will actually be diluted by the other cells that might not be of interest to you as well. So this is something to take note, and it will increase your data quality or your analysis capability depending on what downstream application do you want to do. So there is the target cell isolation from from tissues or blood. So in this case, this is a workflow from solid tissue. So after dissociation from organs or tissue, you get single cell suspension. You can actually put your single cell suspension through our cell isolation kits. So we have a lot of cell isolation kits already made into kits for you for your ease of usage, and you can directly get into your target cells. So for example, in a dissociated tumor here, so for the propionate iodide here is actually the lipid staining, and this is actually your live cells. They are also CD3 positive, which is your targeted CD3 T cells, for example. Yeah, so you can see that it's actually very low number of cells of 3%. However, if you were to do a cell isolation using an isolation kit, you can get up to a 79% kind of an enrichment of your live cells, that is T cells, which is your cell of interest. And you still see there's quite a bit of dead cells. And if you were to put these sample through a dead cell removal kit, you can now actually get up to 90% or close to 90% of live cells for your analysis. So this is actually very useful to increase firstly the quality of your data, and also secondly the data that is actually of interest to you. Yeah, so of course there's also other options. Either you can do it as cell isolation straight from whole blood, PBMCs, or cell suspension. So one other thing that I think it will be very important to take note of is sometimes if you think of just doing like a 5-core spin might get you from whole blood to PBMCs. Sometimes we do see that after a 5-core spin, the stress expression of stress markers or expression portfolio or profile of your cells actually do change a little bit compared to doing a direct cell isolation method. So just keep in mind whatever process you actually put your cells through would might actually have an impact on downstream applications or downstream analysis kind of implication as well. So of course, next is actually the sorting of your cells. So this is the very common kind of sorting methods that you guys would be used to, which is the droplet sorter, where your cells would need to go through the fluidics and be sorted into different tubes. So we actually have our own sorting, which is actually a cheap sorting technique, where it actually is more gentle to your cells. So instead of the traditional FACS sorting method, which is open system and is actually a very it goes through a bit of fluidics and tubings, and it's a very harsh sorting process due to high pressure, the strong decompression when it goes in and out of the tubes, and also the charging of your cells to actually make it drop into the right position, and also a lot of high shear force. This actually stress and kills a lot of cells, especially those sensitive or primary cell lines or primary cells. Yeah. So instead of that, it's actually very gentle, of less than 1 psi. So it's actually very low amount of pressure. And also there's no decompression, no charge applied to your cells. And there's actually very minimal or mild shear forces that's applied to your cells as well. So it does give you a better viability. And of course, you ensure your cells do not undergo any stress changes or profile change in terms of expression. Yeah, of course, it's a closed system, so you can actually work with virus-stained or pathological products or samples. So that's really useful. So of course, coming down to the last bit of your journey or workflow is actually the single cell analysis part. So we actually have a example or experiment that we have done to actually showcase this whole process. So this is an isolation of T and B cells from tumor tissue for single cell immune profiling. So we actually took the tumor biopsy from a person, and then you dissociate the tumor itself. So because we can't process the stuff or the sample immediately. This is actually frozen in cryopreserved, and then we actually thawed it, filtered it, removed for dead cells, and then did a T and B cell isolation, and then put it through the single cell analysis. So this is the data that we have. So for the dead cell removal from the fresh frozen tumor samples, you can see that, because it's frozen and it's from tumor samples, and after processing, we do get very poor viability of your cells. And of course, after putting it through magnetic separation or the dead cell removal, you can actually get up to about more than 80% of your cells that are viable. So that's a very excellent numbers. And next is actually to isolate your T cells and also B cells for your data or your analysis. So this is actually by using the max microbeads. So you can see over here the unseparated bulk cells. So this is actually the total T cells, the CD3 positive T cells. You only get about 40%. So after magnetic separation, you can actually get up to about 80% or more than 80% of your T cells or your cells in this population or sample to actually analyze. And of course, with this flow through, you can also do a B cell isolation for CD19 or CD138, for example, to actually do the B cell analysis. So why do you actually need to do an isolation, and why is it useful? So this is actually a graph actually showing you the top 50 TCR beta clonal types and CD3 clonal types, for example, and it's actually identified comparing the isolated sample or in the bulk sample. So this is non-isolated or non-enriched or non-T cell enriched. So you can see that if you actually use the isolation of your T cells, you can actually see more clonal types here. As you can see over here, there's actually more clonal types, and there's actually a abundance of your T cell CDR3 clonal types for you to analyze as well. So there's actually quite a lot of abundance, and you can actually see more of this TCRs that is actually available instead of if you actually just look at bulk. So if you were just to look at the bulk T cells, then a lot of these TCRs will actually been missed. And if you were to just look at, just the bulk sample, then you will actually also miss out the abundanceness of your individual clonal types. Next is actually looking at the B cells this time round. So again, similar concept, but we're just looking at the top 25 heavy chain of the BCR clonal types. Excuse me. So you can see over here again, this is more pronounced in this case, where in the isolated sample, you can then see the different clonal types that's actually showing up. However, if you're just dealing with the bulk sample, you actually do get a very low abundancy of your cells to actually look at, and you actually lose the sensitivity of all these clonal types that is actually available in your sample as well. So this is something that is very important to take note in terms of preparing your samples and isolating your cells of interest to actually look at what kind of a parameter you are actually interested in and data that you want to work with downstream. Yeah. So with that, I'd like to thank everyone for your time. And I hope this talk has been very helpful for you guys to plan your experiment and the samples that you work with. Yeah.
Oh, thanks, Sam. It's a really, really wonderful presentation. Yeah, we all, really thanks. Even we learn a lot, because most of customers have problem how to dissociate this tissue into single cells. There's many times they look for through the publication. There's no so-called very established protocol for sample preparation. Yeah,
so of course. Yeah, so as I was saying, of course, we are always here to help. Do write into us. We are always very helpful, even if this is something new or we are not, dealing with it. You can always write into us. We do have quite a bit of experts in-house that do R&D and all this process development as well, and we are always happy to help. Yeah.
Yeah, thank you so much. Wait a moment, let me share my screen. Then we almost come to this the end of our webinar. Yeah. So can I see my screen, right?
Yep.
Yeah. So we come to this Q&A session. We come to the end of the webinar also. We may not have time to answer all the questions. So I just choose 2 questions, one for Caroline and one for Dr. Singler. So Caroline, customer is worrying about like, how should I ship my living cells like to another lab, my lab maybe in another country, then how is the best method?
Oh, okay, so if like overseas, then I would say like if you can cryopreserve your cells, that will probably be the best methods, because after that you can thaw your cells and you can still recover pretty good cell suspension with high viability. Yeah.
Yeah. So for Sing, I have a question from our audience. It's about if my cell suspension have a lot of that cells, then I will need to do maybe that cell removal from the kit from Miltenyi, but my cell number is not so allowed. So what's the best recommendation? Yeah, I know the cell removal will, the cell number may be reduced, right?
Yeah. So in my personal experience, even when I'm working with very low quantity of cells, I don't lose too much cells. So I've put through about 10 million cells before, a few million cells before through the kit, and I still do get quite a bit of cells back, like about less than 1 million, I guess, but it's still enough for me to do the 10x genomics kind of a downstream application. And yeah, so I think it's still worth a shot to try. If not, you can always try the instead of the kit, the debris removal solution. In that case, you actually lose less cells, which would be a better alternative as well. So you should try both methods and see which will actually work better for you. Of course, if your cells are not being decreased by too much, if you put it through the kit, then I would suggest the kit, because it does give a better viability percentage as well.
Okay, thanks Caroline and Sing for your wonderful presentation. And thanks again. Then we always come to this end. So finally, I just want to echo again why you should choose our services. We have cost-effective services, very productive, and very fast in TAT lab, which in 1 month you can get results, especially suitable when you are rushed for your one application, paper revision, or any presentation. Yeah. So thanks again for our wonderful speakers, and thanks our audience. Yeah. So, and if you have any question regarding single-cell experiment or sample prep, you can reach us. Also can reach to like Caroline Singh for the sample prep guideline and the best practice. And before we end our webinar, we will have a very short survey. Please help us to finish the survey so we can further improve our future webinar. Thanks again. Thanks. I hope you learn a lot. See you next time.
Thank you.
Bye.
Thank you, guys.